Welcome to those of you who are logging on for today's webinar with Dr. Warner Greene. We will be getting started right at the top of the hour. Welcome to those of you who are logging on for today's COVID-19 webinar. We will get started right at the top of the hour. everyone, and welcome to today's webinar focused on the COVID-19 pandemic. I'm Chris Rebelo, the Director of Communications here at the Buck Institute for Research on Aging. This series is focused on the science of COVID-19 and its impact on public health. And of course, we always try to give you some practical advice to help you navigate your way safely through this pandemic. Today, we talk with Dr. Warner Greene, Distinguished Professor of Translational Medicine at the Gladstone Institute of Virology and Immunology. He is a science guy. His lab is studying how the SARS-CoV-2 virus enters cells, and his team is also screening a library of FDA-approved drugs to identify those that could be repurposed as an antiviral treatment to stop cellular entry and spread of the virus. As the director of the Gladstone Center for HIV Cure Research, Dr. Greene brings a wealth of experience on drug development, and he understands how basic research gets from the lab bench to the bedside. Once again, a reminder, this is a participatory event. If you have any questions for Dr. Greene, please use the chat feature at the bottom of your screen. So Dr. Greene, welcome, welcome. So glad you could be with us today.
Thank you, Chris. Pleasure to be here.
So I think the only way I can start off this conversation is with the word yikes. So latest statistics show that new COVID-19 infections have risen 76% in this country over the past 2 weeks. Last Friday, the Centers for Disease Control said the number of infections in many parts of the country are more than 10 times higher than the reported rate. So are you surprised by this? What's your take on where we are?
Well, we are not in a good place. The infections are spiking mostly through the south and the southwest, even here in California, increased numbers of infections. Clearly, a lot of this relates to opening prematurely and opening poorly, really with the lack of social distancing, the lack of masking. It was not bad. We just can't go back to normal. This virus, we may be tired of the virus, but the virus is not tired of us.
Got it. So late last week, the CDC broadened its warning about who's at risk for developing serious diseases from COVID-19 infection. It suggested that younger people who are obese or have other health conditions can also become seriously ill from COVID-19 in addition to older adults who are at increased risk. What does that tell you about the coronavirus? What do we know now about this virus that we didn't know at the beginning of this thing?
Well, we know it's a very serious infection. While some people, it's like the common cold, that this virus has the capacity to kill and to kill with high efficiency. These, hypertension, diabetes, obesity, there are a number of risk factors, as well as lung disease. I think all of this is telling us that there's a significant endothelial function, an endothelial property that all of these diseases damage or affect the endothelium. And I think this virus, that we're going to see an intimate relationship of this virus with endothelial cells as well.
And so those are the cells that line our organs, right? They sort of provide a barrier between us and the outside world. Is that a good way to put it?
Particularly the vascular system is what I'm referring to. And we know that this virus has a real propensity to elicit clotting in both the arterial and the venous system. How it does that is one of the things that my lab, along with Dr. Katerina Akasoglu, that we are trying to understand how the virus triggers the clotting cascade.
Yeah, well, that's serious. There are young people, older people who are coming down with strokes from this, right?
Correct. Yes, and a lot of young people having strokes that are, just out of the blue with this virus. Got
it. So there have been a ton of studies. It seems like every day there's a new study about this virus. Recent studies show how the coronavirus short circuits our immune system by, in some case, depleting T cells. Now, these papers are not fully peer reviewed, but one described the phenomena as disturbingly parallel to HIV. So can you talk about how the coronavirus, how it impacts our immune system?
Well, I think that we have no immunity against this virus. So when the virus enters our body, the only thing that determines the natural history of that infection is our innate immune response, our primitive. It's a first line of defense. And one of the major factors generating that defense is interferon. And this virus has a way of turning off the interferon response. So allowing it to gain a foothold. And yes, that the individuals who have the greatest degree of lymphopenia are at the greatest risk of developing severe pulmonary disease and ultimately the acute respiratory distress syndrome. Now, the whole interplay with T cells and how antibodies are made, these are areas that are rapidly evolving now, our understanding of them.
Do you think that it will require a cocktail of antivirals and vaccines to really bring this under control?
Well, I think that if we had combinations of antiviral drugs that acted in a synergistic manner, and we could give those drugs early in the course of infection, then such treatments would be highly effective. Indeed, one could even think about using certain types of drugs that were exceedingly safe as prophylactics. In other words, just the way we use antivirals in HIV to prevent, to give them to people at risk, we can prevent people from getting infected in the first place. That would be wonderful if we had those kinds of drugs for SARS-CoV-2, but we do not at this juncture.
So our first question just came in. What about the oral polio vaccine? Would it be worth asking my physician for a dose? And there was actually a story online this morning that somebody is talking about using the mumps, measles, and rubella vaccine as a possible stopgap to provide some protection. Do you have any take on that?
Yeah, well, certainly this is I think your guest previously, Dr. Robert Gallo, may have spoken. I did not see his interview, but he is certainly intrigued and wishes to test the concept that an oral polio vaccine could elicit or induce and stimulate the innate immune response to a point that, in fact, people are more resistant to SARS-CoV-2 infection. I think it's an interesting concept. I don't think that it would be restricted just to the oral polio vaccine as suggested by using other vaccines to try and elicit that response as well.
What about asymptomatic spreaders with all this spike in cases around the country and even here in California? Where are we with do we need to identify those people? How hard is it going to be to identify those people? What percentage of the population out there do you think might be asymptomatic spreaders? That seems like a big issue to me. Is it to you?
Yeah, I think that, well, there's two types of individuals, asymptomatic individuals who will remain asymptomatic throughout the entire course of their infection. And then there are pre-symptomatic individuals who will ultimately develop some type of pulmonary condition, but prior to developing that, they are shedding the virus. And that is that distinguishes this virus from SARS and MERS, where in fact viral shedding occurred late in the course of the infection. That's why public health measures were able to bring SARS under control. Here, the issue is so key. Masking is so key given the asymptomatic or pre-symptomatic phase of transmission. The only way that we can block people from spreading the virus to others is social distancing, which we see is kind of plus minus as we've opened back up. masking does work. It really, really works, and we need to be doing that. That is absolutely essential. If we as a country are going to get by this infection to the point that we have a vaccine, that will be the ultimate solution. But we've got to pull together here and get around get beyond the fact that wearing a mask it's not a political statement, it's a health statement. It's expressing concern for other individuals. I do not want to infect you, therefore I'm going to wear a mask, even though I don't know that I'm infected myself, but I'm going to assume I am, and I'm going to try and protect you. That should be the attitude that we all need to take.
Got it. So another question, just asking me to go back to the question about the polio and the mumps and the measles. Do you think if older people are concerned about their own risk, would it make sense for them to talk to their doctor about getting this vaccine?
Well, well, I mean, I don't think that you can find oral polio vaccine available right now. I think that I think it's not available as I understand it. And this is unproven. I mean, this is hypothesis at this.
Fair enough, yes.
And so I don't know. I think everyone has to make that judgment for themselves. I'm not going out to get vaccinated. I'm using more traditional approaches of social distancing, masking, washing my hands, et cetera, et cetera.
Got it. Well, it just shows how desperate people are to do the right thing for themselves.
Absolutely. And we don't have a lot to really prevent us from getting infected, and that's an interesting concept, but I'd like to see some evidence that it really works first.
Got it. So before we talk about what specifically is going on in your lab, can you talk about this disease, COVID-19? I know when you and I talked earlier, you talked about there's two phases of the infection. How can we understand that?
So yes, exactly. Two phases. The first phase is dominated by the virus, and the second phase dominated by the host. So when people initially get infected and the virus travels into the lung, at that point in time, the virus is in control. It's trying to multiply and replicate, and it's killing epithelial cells in the lung, which is causing respiratory distress. At the point when people get so sick that they need to go to the hospital, that in fact, they have now entered the phase of the disease that is driven by the host. And now that the host is mounting an overly zealous response against the virus, rich in inflammation. And this leads to what's called a cytokine storm, and ultimately to the acute respiratory distress syndrome. So this is very characteristic of many viruses, but coronaviruses in particular, that they elicit this host, that in fact the final phases of the disease and the pathology is driven by the host rather than the virus itself.
Do we have any understanding of why this virus is so very infectious?
Well, we know now that in contrast to SARS, that it has a much higher affinity for its receptor. The angiotensin converting enzyme 2 is the ACE2 is the receptor. And compared to SARS, it binds with about 10 times a higher affinity to that receptor. I think I think that makes it more transmissible between people. And then of course there are the individuals who are super shedders. That have high viral loads when they're asymptomatic and spreading the virus quite effectively. This virus may, a lot of the spread of this virus may be seeded by super spreaders.
Were you surprised that we have not received the summertime lull that some people were thinking that we might get, that once it gets warm outside, boy, the virus is going to fade away?
Yeah, if you look at the coronaviruses, though, about half of them show seasonality and the other half don't. And in fact, summer colds were often caused by the circulating coronaviruses that caused the common cold. I had hoped that we would get a respite, but it does not appear that's the case. I mean, kind of the celebrations around Memorial Day, I kind of watched with great trepidation. The many celebrations of people, it's like, it's over. It's over. The virus is, I mean, no longer a concern. We're out, we're free. We're no longer sheltering in place. And, it was almost predictable exactly what would happen. And I mean, the United States has, we have not handled this virus well. We have the second highest per capita infection rate in the world. Only Brazil, which is in the midst of kind of the first massive phase, has a higher infection rate than us. In contrast, Germany, Italy, France, all of these countries have infection rates which are 10 times lower. Now, and being second in the world right now, we could well be first with the surge that's occurring through the South and the Southwest now, for sure.
Do you think the window has closed for having a strategic, thought out plan to deal with this pandemic in this country?
Well, I think the ultimate solution is a vaccine. And there, the news is so far good. We have 3 companies moving into phase 2, phase 3 efficacy trials. Moderna in July, Oxford and AstraZeneca in August, and Johnson & Johnson in September. And others, many, many others closely following thereafter. We're going to need 4, 5, 6 different vaccines in order, and all manufactured at scale, in order to vaccinate the billions of people who are going to need the vaccine. And I just hope that this does not become the same kind of political. I hope that this process is as equitable and as efficient as possible in terms of vaccine distribution, although I worry that it may not be.
Got it. So let's talk about your lab. I understand that your efforts are really focused on coming up with pan-coronavirals, antivirals. Talk about that.
Well, yes, we're interested in inhibitors that would work against all of the coronavirus family members. It's pertinent to note that there have been at least 3 major entries of coronaviruses into the human population since 2003, SARS, MERS, and SARS-CoV-2. And then, of course, the respiratory viruses, 4 of them. I think 2 of them have entered the human population relatively recently. So this is not a rare event that coronaviruses are transmitting from their animal host, usually through an intermediate. It starts out in the bat, and usually through an intermediate host, some type of animal. In the case of SARS, it was the civet cat. In the case of MERS, it was the dromedary camel. We're not sure about SARS-CoV-2 yet in terms of whether there was an intermediate, but it makes its way into the human population with some regularity. So we can anticipate that in the next 10 to 20 years, we're going to see other potentially pathogenic coronaviruses. So now's the time to actually be creating in antiviral combinations that affect not just SARS-CoV-2, but also getting ready for the next transmission. And so that we are looking at conserved parts of the life cycle of the coronavirus where effective drugs would block the entire family. We're very interested in the approach.
So I understand you were looking specifically at that spike protein that's on the coronavirus. We all see the pictures of it now. I think everybody recognizes the pictures of the coronavirus with those spikes. So what are you trying to do with those?
Red. Aren't they red?
They are red. What are you trying to do in relations to those spike proteins?
Well, the spike protein has 2 parts. One that binds to one part binds to the receptor, and a second part after the virion is taken into the cell via the endocytic pathway into vesicles. And then once it gets into the what's called the late endosome or lysosome, that's where it fuses. That's where it melts its membrane with the membrane of the lysosome and actually escapes into the cytoplasm. We're trying to block that fusion step. And that fusion domain is conserved amongst almost all of the coronaviruses. So if we can come up with a fusion inhibitor against SARS-CoV-2, it should be active against the other coronaviruses as well.
And then I understand you're looking at some FDA-approved drugs as potential therapeutics, antiviral therapeutics. What's that about?
So we certainly were not alone in this. In fact, a group at Sanford Burnham in San Diego screened what's called the Reframe Library, which is the collection of all of the FDA-approved drugs, as well as all drugs that were shown to be safe in humans in phase 1 testing. And they were able to identify about 30 different drugs that are used for other diseases that had antiviral properties that were able to block the replication of this particular virus. And now the question is, can we combine those drugs and make even more synergistic? Can we make a synergistic therapy? Could we combine one of those drugs with the fusion inhibitors that we're coming up with and see some type of synergy as well? For example, remdesivir, which we all know, unfortunately, remdesivir is only administered intravenously. It's the Gilead drug. It can only be given intravenously. And so as we were talking about earlier, what you'd really like to be doing is giving remdesivir before people ever come to the hospital in the first place.
Right.
When the virus is in control of the pathology. So we need drugs, for example, if they could come up with an aerosolized form of remdesivir or a subcutaneously administered form of remdesivir, that would be incredibly valuable because you could use that as an outpatient. And remdesivir is a drug that should have activity against almost all of the coronaviruses. We know it's active against SARS, we know it's active against MERS, because it's affecting a key RNA, the way that the virus reproduces itself.
Got it. And then I understand you're also working on a project with Melanie Ott, who was an early guest on this series,
Sure.
where you're looking at organoids. Is that true? Lung organoids? What's that?
Melanie has developed a lung organoid system, and we are very interested in what's the mechanism by which this virus kills the lung epithelial cells, the type 2 pneumocytes, the cells that are primarily making the surfactant in our lungs. We're interested in how that killing occurs and what kind of programmed cell death pathway is activated. And so we will be using lung cell lines to begin with, but then once we have kind of get data there, we'll then translate it into the lung organoid system that Melanie's laboratory has developed.
So having organoids, I would think that's a much better model than just sort of looking at just things in a cell in a dish, right?
Yeah. Well, these are in a dish too.
I understand, but they're more complex. They're the tissue, yeah. Right?
They're much more. They are a much better representation of the actual organ in vivo than, for example, cell lines are. So yes, it's a clear advance in terms of testing for sure.
So let's talk about vaccines. There's a lot of activity.
Yes.
Who are the major contenders? Like what type of vaccines are likely to be developed? Where is that state of the science?
Well, I think at last count, I think there's something like 170 different vaccine efforts around the world. And these range all the way from making the earliest types of vaccines where you take the virus, you kill it, and you use that as the vaccine. So a killed vaccine, that's being developed. there are also efforts to make an attenuated form of the SARS-CoV-2, kind of a live but attenuated form of the virus. That's, for example, the kind of vaccine that Sabin developed for polio, in contrast to the killed vaccine that Salk developed. But more advanced approaches are now looking at subunits of the virus. For example, the spike protein, which we've talked about, is a very popular target for vaccine development. Again, the idea being to elicit antibodies that block the ability of the SARS-CoV-2 spike protein to bind to its receptor. Neutralizing antibodies would be the kinds of antibodies that these vaccines would be aimed to produce. So I'd say that the spike protein is the target of most of the vaccine development, but there are broader efforts as well. Not all of our eggs are in one basket, for sure.
But do you think once we get a vaccine, will it be in one of those baskets, or are we going to need different baskets to respond to COVID-19? And if there are, how the heck does a person, or does a doctor decide, or a health system decide, which vaccine am I going to give to my patients?
I heard it said yesterday, you don't necessarily want the first vaccine, you want the best vaccine.
Right.
The best vaccine might differ depending on your age. It might be necessary for people who are at higher risk, who are more elderly. They may benefit more from a particular type of vaccine, whereas younger people, a different vaccine may be the most effective. I mean, these are all questions that the answers to which we don't have. But what we do have is an unprecedented effort going into vaccine development. And of course, these vaccines are being manufactured at risk. So not even knowing whether they're going to work yet or not, they're scaling to hundreds of millions of doses of these vaccines in the anticipating rapid deployment should they get a strong efficacy and safety signal.
So given that, I know there have been different predictions. Gee, we might have a vaccine by the end of the year. We might have one by next spring. It seems like there needs to be, for safety and efficacy, there really needs to be a lot of work done on large groups of people so we do get the best and safest vaccine. Do you have any sense of, are you at all concerned about safety studies, or what's your idea of when we might actually have vaccines available?
Well, I think the speed of safety testing and efficacy testing is somewhat determined by, certainly efficacy is somewhat determined by how much, where you're testing. If there's a lot of virus in the group of individuals that you're testing, then you can generate an efficacy signal much quicker than if the virus is rare. For example, that was part of the problem with the Ebola vaccine that was developed initially, that they almost got there too late to understand whether or not the vaccine was working, because the Ebola epidemic was dying down rapidly. I don't think there's going to be any lack of virus in the community. So I think testing will proceed at pace. But you're absolutely right. We cannot cut any corners in terms of safety. And again, I just want to, I think we all should be concerned that this is not a political issue. We are moving quickly to a vaccine, but there should be no connection between the development of of effective vaccines and the presidential election.
Got it. Yep. So there was a question that came in about back to super spreaders. How are they going to be identified? How can we identify them? And then that might lead into a comment about antibody testing. I don't know, but how do we find these people who, unwittingly are out and about and might be infecting others?
Yeah. So if they're wearing a mask, they won't be infecting others. That would be good. It comes back to the kind of surveillance testing that we really, and the first line of public defense is that you do widespread testing to identify individuals who are infected, and then you contact trace, you trace all their contacts, and then you quarantine the contacts and you isolate the individual who was detected as infected. We are not doing that in a systematic way in the United States. And I think that to a great extent, that's one reason that we're not gaining control of this virus. We don't know where it is, and it's undergoing community spread now. I mean, we're going to see, I predict, an overwhelmed hospital system in Florida, perhaps in Texas, and in Arizona. I mean, the kind of, it may be uglier than what happened in New York City.
Wow.
And yeah, I mean, the next 2 to 3 weeks in those states, it's not going to be pretty at all. We had a second chance, by going into the lockdown. Virus decreased in the community. We had a chance to come out of the lockdown by doing a lot of surveillance testing and finding the infected, removing them and their contacts from society so society could continue to work. But we opened up at a time when virus was still rampant in many states, and we opened up in a non-safe way. So we are, we, in contrast to most of the developed countries in the world, for example, Germany, France, Italy was hit so hard that their rate of infection right now is 0.5 per 100,000, and in the United States, ours is 10.7 per 100,000. So 20 times more virus in the United States than Italy. We're just not dealing adequately with preventing the further spread of this virus.
So a question just came in, which is a perfect follow-up. Because there are so many questions about the virus and how infectious it is, what about the opening of schools? So schools are planning on opening somehow in the fall. Do you believe that public schools are going to be able to keep students and their teachers safe?
Well, I've heard interesting comments about this from George Rutherford, who's an epidemiologist at UCSF, and he's reviewed a fair amount of data now are emerging from places like Iceland, I think Spain, China, et cetera. And for children less than 10 years of age, they're not heavily infected. The way they get infected is they get infected from their parents or possibly from their teacher. Once infected, they don't spread it to other classmates. And they don't tend to because of that, kids are not bringing the virus home with them. So this is exceedingly good news for, elementary schools, et cetera. Now, the whole thing changes once they reach puberty. Then, adolescents then act like adults in terms of infectivity. So you could make an argument that you could open up schools, and the people that you need to be testing frequently are the teachers. And you need to be, and the parents, the parents need to be, because they're the source of the virus that will infect the children. So I think it's good if that's not well recognized yet. But if those data are confirmed, I think it's very good news for opening up at least the elementary schools and preschools in the future.
Interesting. Okay. I've got another question about whether you think there's going to be a surge in the fall, and somebody's going, what can I do to protect myself once people are forced to spend more time inside? Because now it's come out, if you're outside and there's a good breeze, you're probably pretty safe. Go outside, enjoy the outdoors. But that's going to change in the fall.
Well, I think that I think that we will probably see another surge in the fall. It'll be accompanied by influenza as well. I think the one thing that everybody can do in the fall to help protect themselves is to get an influenza vaccine. And we won't by the fall have a SARS-CoV-2 vaccine. It's also not, the other point I was going to make is it's not clear how long the durability of a vaccine will last. For example, the flu vaccine, we have to change it every year to match the virus that's circulating. Sometimes we get it right, sometimes we don't get it quite right. Hopefully, the SARS-CoV-2 vaccine will last for a number of years, but it's possible that we may have to get vaccinated more frequently. It probably will not be like a measles vaccine where one vaccination lasts you for a lifetime.
So I got a question from somebody who is interested in antibodies. So if somebody has survived COVID-19, hopefully they have some antibodies. Nobody knows how long those antibodies are going to be good for. But is that going to impact who needs to get a vaccine or how somebody might respond to a vaccine? Is that going to be part of the larger picture as well?
Well, for people like you just described who've survived COVID and have good antibodies in their plasma, I would, if they are willing, I would suggest that they contact their blood bank and consider donating plasma, which could be convalescent plasma, which could then be given to people who are really quite ill. The blood banks are coordinating this type of convalescent plasma administration throughout the country. That will ultimately lead the fact that there are such antibodies from recovered people. We will ultimately have now monoclonal antibodies from the best of those producing clones of cells. And those will become a very valuable therapeutic. That's kind of the bridge, convalescent plasma, monoclonal antibodies, all passive immunity. You're giving passive antibody protection, which only lasts as long as the antibody survives. And then the next step, of course, is active immunity, the vaccine.
Got it. So somebody wants to know who will decide how a vaccine is deployed to the population and in what order? Who's going to be in charge of that part of the deal here?
That's a very good question. I would have thought that it's either going to be HHS, Health and Human Services, or CDC. Those would be my two guesses. Probably HHS will be in the lead.
There was a news report this morning that shows that perhaps the virus is mutating, or that there is a mutation in part of the virus that is making a particular version of it more infectious. There was a question about mutation. Is that going to make it more difficult to find a vaccine? Is this virus changing in front of our eyes?
So it's an RNA virus, and by definition, the RNA viruses change more than DNA viruses. That said, SARS-CoV-2 and the coronaviruses, they all have this proofreading enzyme, which tends to correct a lot of the errors that are made. So they do not tend to mutate as much as, for example, HIV or hepatitis C. So that's good news for vaccine development, that the sequence, while modestly changing, is not radically changing all the time.
So we are getting some questions from folks about how to live in this time of semi-opening up. You can do a few more things, but not everything. So before I get to one of those questions, I want to talk to you about how your own family made a decision, basing risks and what's going on. Talk about that, because I think I love the thought process that went into it. So talk about that.
Right. So I have two daughters and three granddaughters. One daughter is a nurse at Lucille Packard Pediatrics, and the other daughter works in Los Angeles. And so we have not seen our grandkids in person for many months. And it was a special occasion, a very special birthday for my wife. And so we decided that we were going to meet halfway between Los Angeles and San Francisco. We would rent a house, and my daughter got tested. Her husband got tested. Also, the LA granddaughter was about to go back to preschool. And so we thought it was a good thing to get together before she went back to preschool. And so we got together and we decided that all of us had been following had been, socially distancing, not exposing ourselves, et cetera. So, and Allison, who was kind of the wild card and because of the nursing, was tested negative. So we decided to interact normally. And that was a calculated family decision that we all undertook, and it was the most wonderful weekend imaginable. But now we're back. Now we're back to the social distancing, because the girls are seeing other kids a little bit. There's people are starting to form pods, and so their social network has expanded, but then they're trying to stay within that pod. But Peggy and I, as the pods have increased, Peggy and I have become more conservative again in terms of our interactions with the grandkids.
Got it. So there's a question here. If we hire a live-in nanny for newborn care, how long should she and us be wearing masks when interacting with each other and when she takes care of the baby, assuming either one of us could be asymptomatic and spread the virus for a while? So when you have situations like that, how long a lead time do you need before all these people who are in the same space are safe?
So in general, the quarantine period for someone who is of unknown infection status is 14 days. And that's set because 95+% of all symptoms will develop within, people who are going to become symptomatic will do so within that period of time. It's not perfect, but it's pretty good. So now it's a live-in person. You said to live in, okay. So I think certainly, quarantine, this type of 14-day quarantine can now be replaced by testing. You can test the individual. If they test negative, then you don't really need to kind of do this 14-day. So that's what I would do. If I would get everybody tested, and everybody's negative, then you're good to go. But that only works to the point that those 3 adults with the baby, that they continue to socially distance themselves, and observe appropriate public health measures. Otherwise, any one of them could become infected from someone on the outside.
So I got a follow up to our discussion about schools. Somebody would like to know, how much greater the risk is it in a young person who is in puberty and adolescent? And should these kids and teens be kept out of school longer? So how should you have any sense of how the school should deal with older kids?
Well, I think high schools are going to be different. I think it's going to be more difficult. But again, I'm not an expert in terms of public education and how best to respond to this. I do know that I think the homeschooling, from what I understand, is not going well, that it's very difficult. And I think that we need more creative approaches to the education of our young children, because this could go on for, we could be in this situation for an indeterminate period of time. I hope that the vaccines are fantastically effective and are deployable within a quick period of time. But in all reality, we probably are looking at another year of virus circulating in our communities and having to do some type of public health interventions to keep that virus down. And I think it just emphasizes we have not figured out how to live with this virus. We're not very effective at it. Other places in the world seem far better at it than we do, but we need to get our act together. But I don't know how to answer the high school question.
Got it. So somebody asked a question about the experience in Sweden. So Sweden did a thing where they kept everything pretty open and they were looking for herd immunity. How's that worked out?
Not good. They've had a lot of, I was just looking at Sweden, infections are on the rise in Sweden again. They had a dip, but now it's on the rise again. They've had, if you compare to Finland, Norway, and Denmark, a lot of excessive death. In fact, the public health minister whose idea this was to try and create herd immunity, he ultimately had to step away from that position and to implement, less, some public health interventions had to be made. Also, unfortunately, they didn't come anywhere close to herd immunity. I think that their antibody positivity is at somewhere around 20% in the population. I think that's what I read. So that's a failed experiment.
So among all of this kind of nightmare of COVID-19, has there been any silver lining for you personally or for the field in general?
Well, I think that as a virologist, I could never have imagined a virus bringing the world to a full ground stop like SARS-CoV-2 did. When SARS-CoV-2 broke out, when COVID-19 broke out in China, initially I thought it's going to be like SARS. There's going to be a few thousand, but then it grew and it grew and then it jumped countries and then it spread to Europe. And we're facing the largest public health challenge in the last 100 years since the great influenza or the Spanish flu. So one thing that's been gratifying is to see the incredible scientific collaboration that has occurred. Scientists really kind of dropping, really working in ways that are not in their own self-interest, but in the interest of progress in the field. And that's been very, very satisfying.
That's great. So given the current lack of coherent leadership, is there anything in addition to wearing masks that people can do now, either for themselves or for their fellow citizens? Are you like here in California, it's getting easier to get tested. Do you think everybody should go out and get tested? What can people do?
Well, I think the wearing a mask is key. I think many countries in Asia, they faced respiratory viral outbreaks, and masking is a routine part of their response. And for example, South Korea brought it under control, Singapore, Hong Kong, and masking was a big part of that. Unfortunately, masking now has become a political statement in the United States. And we have just got to stop that. If there was anything that should have brought this country together, a common threat, it was this virus, COVID-19. And I hope that at some point, we can stop this blue versus red issue and really face this virus in a consolidated, unified manner. But that requires leadership. That requires leadership at the top to do that.
So So I always get questions from folks who are following the rules, staying at home as much as they can, physical distancing, wearing masks, but people also want to know what they can do to give themselves the best shot of doing well should they become infected with this thing. So do you have any advice for people about how to get a strong immune system? What should people be doing? Is there anything that people can do?
Well, if I got infected, I know the first thing I do is try and get subcutaneous lambda interferon. There's a trial underway at Stanford. I'd try and get enrolled in that trial for sure. You really want to be giving the antivirals early in the course of the infection. Unfortunately, you can't give remdesivir because of the intravenous. And also there's very little remdesivir around to be administered. It's very difficult to synthesize it. So no, there's not a lot. There's not a lot that we can do. The studies with dexamethasone were promising, but of course that's modifying the host response. That's for the very seriously ill, but that was good news that dexamethasone works. And I think we're going to have a variety of additional antiviral drugs that are going to be becoming available, or identified and then become available. I think there'll be some repurposing. But for example, the favipiravir, which is the Fujifilm kind of version of remdesivir, that can be given orally. And so it'd be wonderful if that drug actually gets better developed, tested, and deployed. But right now, we don't have a lot to give.
Got it. So there's a question for here locally. So Marin County is home to San Quentin Prison.
Yes.
There's a total of 832 active COVID cases at the prison. What is that a danger to the larger community?
Yes, to the extent that the guards become infected and the guards introduce it into the community. But congregate living situations like nursing homes, prisons, businesses where people work in close proximity, like meatpacking industry, et cetera. I mean, some of I also have seen some of the Amazon warehouse workers have become infected. These are all situations that require special interventions. Like in the nursing homes, for example, the state of New York is now testing every nursing home worker every week to make sure that they're negative, and they know that all of the people in the nursing home are negative. So you've got to take special approaches in those types of congregate living situations. And yes, Marin is kind of a hot spot right now, but it's mostly driven by the prison outbreak.
So let's talk about older adults as we get close to winding this down. Obviously, that's the Buck's focus is on older adults, keeping them as healthy as long as possible. Is there any sense of why this virus impacts older adults, that they're more at risk for serious complications? And how is that risk factor gonna work as far as upcoming vaccines and potential antivirals?
Well, I think that older people in general are more likely to have the risk factors associated with more serious disease. They may also have a less, their immune response, a certain degree of senescence in their immune response. Although you might think that could help in the situation when during the host phase of the disease, you would think that, well, a weaker immune response might mean less chance for cytokine storm, et cetera, but that doesn't seem to be the case. Yeah, I think that it's probably just an overall kind of lack of innate immunity or diminished innate immunity, so the virus can get the foothold. yeah, that would be my guess.
So when you talk to your colleagues and collaborators, what are, as far as scientific possibilities, what are you mostly excited about? Like, where do you go, wow, that looks really promising? Is there any particular effort that you're like maybe willing to put a bet on, or what's most promising?
Well, I am very confident that science will prevail. I do believe that we're going to have combination antiviral therapies, and I think we're going to have safe and efficacious vaccines. But I think that all of that will require some time, and that's the issue. Can we stay healthy? Can we, as a country and as a world, stay healthy and learn to live with this virus until we can get rid of this virus, or have ways of really managing it? So I'm impressed with the amazing focused effort that's going on around the world. I don't think we've ever seen science so squarely focus on a problem like this as we are seeing now, and it's going to yield great fruits. We just, we need the gift of a bit more time. So people need just to, do not yield to COVID fatigue. Hang in there, and science will get us through this.
That's the perfect way to bring this to a close. Thank you so much for taking the time to be with us today. I also want to thank everyone who participated in today's webinar. We got some great questions. A video of today's event will be posted on our extended session website in the near future. So our next webinar happens on Wednesday, July 8th at 11 a.m. Pacific, 2 p.m. Eastern. I'll be talking with Dr. Susan Desmond-Hellmann, the former CEO of the Bill and Melinda Gates Foundation. We are thrilled that she's making time to take part in this series. Dr. Desmond-Hellmann is an expert in public health. She has dealt with earlier pandemics. As former chancellor of the University of California, San Francisco, she also understands the ins and outs of academic research and has dealt with healthcare systems and patient care. And if that's not enough, as former president of product development at Genentech, she understands how drugs are designed and how they get to the marketplace. So we will have a lot to talk about. So I hope that you enjoy what I think might likely be a more subdued July 4th holiday coming up. Please stay safe, stay healthy. I will be back here on July 8th, and I hope you will be too. I'll see you then.
Thank you.